So at this point in the chapter, we have talked about all four stages of cellular respiration, aerobic respiration specifically, and we're going to bring back a topic from chapter 6, which is feedback inhibition. So you may recall when we first learned about metabolic pathways, about enzymatic pathways or biochemical pathways, which are series of enzymes that act in a sequence, that there was this mechanism called feedback inhibition by which the final product of the whole pathway could inhibit some earlier enzyme in the pathway. Now, the inhibition is not permanent, but it acts as a sort of a thermostat in the cell, such that the cell isn't making too much of something or too little of something. And so what happens in cellular respiration is there's actually several points where there is some feedback inhibition. And this diagram shows you several of them. But rather than memorizing them specifically, what we want to look at is ATP actually acts as an inhibitor for, if you look here, ATP, which is the product of cellular respiration, that's the energy currency that the cell is building from ADP and ATP using the energy from the bonds of organic compounds. So ATP actually acts as an inhibitor to one of the very early enzymes in the first phase of respiration called glycolysis. So that's the name of the enzyme is phosphofructokinase, but it acts as an inhibitor towards that early enzyme. And so when there's too much ATP being made, it can shut down glycolysis temporarily, not permanently. And then when the levels of ATP start to drop, then that inhibition is removed. And you see there is another point of inhibition that's not particularly shown. Actually, it is. I can't see it because of my computer. It's shown over here. NADH can inhibit that pyruvate dehydrogenase, which you'll recall is the enzyme that catalyzes the one step of pyruvate oxidation. And so you have these different steps that are inhibited by some of these products. And so it's a good example of feedback inhibition. So up to this point, we've been talking mostly about aerobic respiration, but I had mentioned in one of the early lecture parts that we were going to come back and talk about anaerobic respiration. And so we will briefly. Anaerobic respiration has to regenerate NAD+. It does have an electron transport chain and a final electron acceptor, but the final electron acceptor is not oxygen. So just a couple of examples. Methanogenic archaeans use carbon dioxide as their final electron acceptor, which makes methane as a product. And methane is released from cows, and it's actually kind of a pollutant, especially in areas where a lot of cows are raised. But it does go into the atmosphere as a pollutant. The green and purple sulfur bacteria, that's two groups, the green sulfur bacteria and the purple sulfur bacteria. But both of those types of bacteria use sulfur, hence the name, as their final electron acceptor, and it produces something called hydrogen sulfide. So you have these alternatives, these organisms that can use one of these alternative electron acceptors. And so anything that fits that description would be called anaerobic respiration. Another type of respiration that I mentioned earlier in one of the lecture segments is fermentation. And fermentation does also regenerates NAD+, but it does so without using an electron transport chain. And so you'll recall that the electron transport chain in aerobic respiration was responsible for a large amount of our ATP yield. So any cell that doesn't use the electron transport chain is going to get a much lower yield. In fact, in this case, you're only going to get that 2 ATP that is generated net in glycolysis. There's a couple types of fermentation that occur. Yeast do what's called ethanol fermentation. Ethanol is a product. It's the type of alcohol that you can drink. And really for the yeast, the ethanol is a waste because the yeast is just trying to regenerate its NAD plus, and the ethanol is just the byproduct of that. Now, for humans, ethanol is a product that we can sell and consume, but for the yeast, it's their waste. Also, as a byproduct, you get some CO2 produced. And so those two things, CO2 and ethanol, are really not what the yeast are going for. They're really trying to regenerate their NAD+, but as a side product or a byproduct, they make these other things. So animal cells can also do fermentation, but animal cells don't do ethanol fermentation. Our cells do lactic acid fermentation. And so this happens in your muscles. If you exercise too vigorously and there's not enough oxygen coming in, then your cells can temporarily switch to a lactic acid fermentation process until enough oxygen can be brought in. And that produces lactic acid and NAD+. Remember, NAD+ is the goal, to regenerate the NAD+. And so lactic acid is the product in that process. And I put this little miniature diagram from earlier because remember all these purple parts, so it was all fermentation. There's no electron transport chain and no oxygen. We used glucose in our description of respiration. We talked about glucose being broken down, the oxidation of glucose, to get enough energy to synthesize ATP from ADP and inorganic phosphate. But other nutrients can be used. And we think of nutrients, we want to think of those monomers that are the monomers of our biomolecule categories. And so I give you some examples here. Amino acids, I give you kind of a general structure of an amino acid that we've seen before. Amino acids can be deaminated. That means removal of this amine group, this amino group. And the rest of the amino acid is a 2-carbon structure, and it can be converted into molecules that can enter in sort of a side entry into glycolysis or into the citric acid cycle as acetyl. And then fatty acids are typically broken down into 2-carbon groups, which you'll remember are acetyl groups, which then can be modified to acetyl-CoA, and that can enter into the Krebs cycle. And so all these other molecules that you have as food, as nutrition, what you're really bringing in are molecules that can be modified and then put into some point of cellular respiration in order to extract from those molecules their covalent bond energy in order to transfer it to ATP. This is a map, kind of an artist drawing of what all the molecules can do, and it's, as you can tell, it's kind of complicated, but if you look at it, the macromolecules, nucleic acids are broken down into nucleotides, proteins are broken down into amino acids, polysaccharides can be broken down into monosaccharides, and then lipids and fats can be broken down into fatty acids. So from this point, this is where if the sugar is glucose, it can be immediately used in glycolysis, or it can be converted into one of the molecules of glycolysis. If the amino acids are deaminated, they can be fed into different points of this process, and same with fatty acids. So it's the same as I was just showing you. And then nucleotides, to be honest, your cell does not rely on nucleotides as a main source of energy in that sense. It doesn't it does, but only at last resort will your cells oxidize those. So we're mainly talking about the amino acids, the sugars, and the fatty acids. And it may be why nucleic acids are not a major food group, but proteins are, and carbohydrates are, and fats are, because those bonds, those covalent bonds, get you the energy that you use. And I added in this one, this is a metabolic map of the whole cell, not just cellular respiration, but everything that goes on in the cell. And the artist has drawn this like a subway map. I've seen this kind of map done many ways, but this one is kind of interesting because it's a subway map. But what you notice is most of the stops, so to speak, in this subway map, most of the metabolic stuff is happening around the citric acid cycle. So you see a lot of things feeding into and coming off of that. And that's why we learn about cellular respiration in our beginning biology class, because it is such a central key part of the cell's metabolism. You do not need to memorize this. In fact, if you memorize this, I think you'd be ready for your PhD. So that is the end of chapter 7, and you may need to go through it again. You can always turn up the speed a little bit, slow me down, pause me, rewind me, and make sure you get the details and go into your post quiz and see how well you're prepared.